students, this is Raheema again and welcome to the last video of reproduction in animals, where we are going to solely focus on the modes of asexual reproduction. Okay, so let's start without further ado, and for that I'm going to share my screen with you people. So the topics that we are going to discuss include budding, binary fission, multiple fission, spore formation, cloning, fragmentation, and lastly, we are going to finish with regeneration. Okay. So the first is process is of budding. Budding is the process where a tiny little outgrowth develops on the parent's body. This outgrowth then grows more and more, and finally it it buds off. That means it gets cuts off from the parent's body and develop into a new individual independently. Okay, here budding in yeast is shown. Yeast is a unicellular fungus. Okay, so you can see this is the parent yeast cell. This is the parent yeast cell. It is forming a bud, which is here. Okay, now the cytoplasm starts dividing. Bud is with a nucleus. Okay, and then it has separated as a separate cell. Okay, and more yeast cells are formed when there is a chain of bud that forms over the cell that first budded off. Okay, this is how the yeast cell grows. Okay, and in the next picture you can see budding is shown in hydra. Okay, in the hydra also you can see a tiny little outgrowth in the form of bud. This bud also it grows just like the parent. Okay, it finally grows off so that it can get cut off and develop into a completely new individual. So whenever you are asked what is a bud, you can simply answer that is just an outgrowth on the parent's body. Okay. Let's move on to the next process, that is of binary fission. Binary fission is a simple process where one cell divides into two. Okay, here binary fission, I'm going to write the definition also. Binary fission is the process of one cell dividing into two daughter cells. Okay, here the binary fission is shown in case of a bacteria where the bacteria nucleoid elongates first of all, and after the nucleoid divides, the cell wall and the membrane they begin to form a transverse septum, which is here. This transverse septum it covers the whole length of the cell, and finally the cell divides into two daughter cells. Okay, we will look at the binary fission, the three types of binary fission in other unicellular organisms, which are protists. Okay, first is longitudinal binary fission, where the plane of division is longitudinal along the length of an animal. Okay, second is transverse binary fission, where the length of the division is along the horizontal plane. Okay, which is this. And in irregular binary fission, there is no plane of division, okay, which occurs in. Can you identify the animals? The first animal is Euglena. The second one is Paramecium. and third one is amoeba. Okay, the division is same. One cell is dividing into two cells, but the division plane is different. The plane of division is different. Okay, so this you have to remember. Euglena longitudinal binary fission, paramecium transverse binary fission, amoeba irregular binary fission. Okay, then there is another type of fission which is multiple fission. Here one cell divides into multiple daughter cells, okay, which is very common in malarial parasite, which is plasmodium. Okay, this is how it is able to spread the infection in human body. Okay, you can see the nucleus dividing into two, then which further divides into four nucleus, and it keeps on dividing, and the cell it releases, a cell bursts and it releases the multiple daughter cells. In case of plasmodium, these multiple daughter cells are known as merozoites. Merozoites are uh, responsible for spreading the infection. Okay, let's move on to spore formation. Spore formation is common in fungi like bread mold. Examples of bread mold is rhizopus, mucor, etc. Okay, and penicillium is also an example. Aspergillus is another example where the reproduction will be the same. Okay, that means by spore formation, but the types of spores will be different. Okay, but our concern is the formation of spores, how reproduction is taking place with the help of spores. Okay, so in this picture you can see the fruiting body inside which the spores develop is known as sporangium, which are borne on erect hyphae known as sporangiophores. Okay, this is just a structure of a hyphae. Okay, a mass of hyphae is known as mycelium. So this is a mycelium. Okay, I'll just write also the thread like structure is known as hyphae. Okay, and mass of hyphae is known as mycelium. Okay, so this is a mass of hyphae and this is just one hyphae. Okay, rhizoids are the root like structures which actually absorb the nutrients from below. Okay, which you can see in this picture also. In this picture also it is very visible. The spores are tiny little fruiting bodies, okay, which contain the nucleus, which develops into, which contain all the genetic information which develops into a new individual. Okay, these, when the sporangium bursts open, these spores they can travel to longer distances with the help of wind, air, and that is why within a small period of time the fungi is capable of spreading very fast. Okay, and if the condition are conducive, like if there is a right amount of humidity, right amount of temperature, heat, warmth, and everything, then the fungi will spread faster. Okay, all this is because of spores. Okay, spores, don't forget, spores are the fruiting bodies, tiny fruiting bodies, and they are capable of undergoing dormancy also, that is the stage of resting. They rest if unfavorable conditions are there, and when the favorable conditions return they break dormancy and start developing into new adults. Okay, so let's move on to the next process, that is cloning. Cloning is the process where identical uh, individuals are produced. Okay, so it is the process of producing identical organisms. Okay, in this particular picture, I'm going to explain I'm going to explain how Dolly the sheep was produced. Okay, so a black faced sheep was taken as a donor for the egg, and the nucleus of the egg was removed. Okay, and a white faced sheep was taken for donating a somatic cell, any vegetative cell from its body, and its nucleus was intact. Okay, and these two cells, they were fused with the help of electricity. Okay, so this was an egg fused with the cell. Remember that the cell containing the nucleus is of the white faced sheep. The nucleus is of the white faced sheep. This was induced to form an embryo, and this embryo was implanted into another black faced sheep, which acted as a surrogate mother. And a black faced sheep with white faced lamb, that was a clone of the white faced sheep, was produced. Okay, so the clone of which sheep is produced here? The white faced sheep is produced here because the nucleus of the white faced sheep was used for the production of the embryo. Okay, the donor of the egg and the surrogate mother, in this case both are black faced sheep, but still the lamb that was produced, it was the white faced lamb. Okay, because the nucleus was of the white faced sheep. This is how the cloning process is done, and this is how Dolly the sheep was produced. Okay, I'm going to show you a short video on the process of cloning. I hope you enjoy it. So let's start. Okay, so we are going to begin the recording in a moment. Okay,
Spot the two
so
clones. When we talk about clones,
let's start the video.
spot the two clones. When we talk about clones in science, we mean organisms that are identical copies. They have the same DNA as each other. Identical twins are examples of naturally occurring clones. Both plants and animals can be cloned. Let's start with plants. A gardener, farmer, or plant breeder might want to make many copies of a particular plant quickly. The easiest and cheapest way to do this is to take cuttings. Shoots are cut from the parent plant, then the end of each shoot is dipped in hormone rooting powder and placed into a pot of soil. The hormone rooting powder will encourage the cutting to start growing roots, and soon a whole new plant will have grown, which is identical to the original plant. The method works because the shoots of the plant contain stem cells, which are able to differentiate to form different cells and tissues. You can find out more about this process by watching this video. However, you might want to produce hundreds of plants from only a small piece of plant tissue. To do this, another method called tissue culture, also known as micropropagation, is used. Here are a few cells taken from the parent plant and placed onto a nutrient jelly using antiseptic technique. This means making sure no microorganisms contaminate the jelly. The cells will start to differentiate and form new plants. Animals can also be cloned, but different techniques need to be used. In animals, only embryo stem cells have the capability to differentiate all the different types of cells found in an adult. There are two methods you can use. The first is called embryo cloning. A farmer might have a cow that gives a lot of milk and wants to use her to create many calves. The cow is artificially inseminated using the sperm of a boar. The embryos grow until they form a ball of embryonic stem cells. Before they become specialized, the embryos are removed from the uterus, divided up into separate embryos, which will all have the same DNA, and then each is placed into the womb of a different cow. These cows are surrogate cows. They are just being used to grow the embryos until they are born. The calves will be clones of each other, but not a clone of the mother due to the bull's sperm. Hopefully, the calves will have the characteristics that the farmer desires, but as sexual reproduction is involved and the genes from both parents are randomly mixed, there is always the chance that they won't. There is a way to get around that problem, and that is to use another technique called adult cell cloning. This is used to form a clone of an adult animal. You might have heard of Dolly the sheep. She was the first mammal to be cloned back in 1996. There are several steps to this process. An unfertilized egg cell is taken from an adult female, and the nucleus is removed. A body cell, such as a skin cell, is taken from a different adult. The nucleus is removed from this adult body cell and is inserted into the egg cell. An electric shock stimulates the egg cell to divide to form an embryo. These embryo cells contain the same genetic information as the adult body cell. When the embryo has developed into a ball of cells, it is inserted into the womb of an adult female surrogate to continue its development, so 3 different adults are used. The baby born will be a clone of the adult that donated the body cell because it has the same DNA. This technology is very controversial and raises many ethical issues. One worry is that it could be used to clone humans. Questions raised by this might be, how many embryos would be destroyed in order to successfully clone a person, and would the clone have the same human rights as others? So in this video, we have looked at what a clone is, and the different methods used to clone plants and animals.
This was a very informative video, and it explained cloning not only with respect to the animals but
Can you
also,
spot the 2 clones?
sorry,
When
with respect to the plants. Okay, cloning is a very good method with respect to plants because it will increase the production of higher yielding varieties of crops. But for animals, it raises many ethical issues. We can get into it later. But let's continue with the next process, that is fragmentation. Fragmentation only takes place in filamentous forms like algae, and one such algae is green algae is Spirogyra. Okay, Spirogyra has filaments like these, and a septum develops. Tiny little septum can develop in the filament of the Spirogyra, which divides it into two, and this process is known as fragmentation. These fragments then can develop into new individuals. Okay, very simple process it is, but you have to remember that it is very common only in the filamentous forms. Okay, let's move on to the next process, which is quite interesting. Some people, they think that regeneration can also be regarded as fragmentation. Okay, it can be regarded as fragmentation. There is nothing wrong in saying that regeneration, where the body gets fragmented, it and each part of the body can divide and develop into a new individual. Okay, but this process, that is regeneration. Okay, here we are talking about animals who get accidentally divided. Their body gets divided into two parts, and then after that, what? Will the animal die? No, the animal will not die. Okay, each part of the body, because this is an accident. Okay, so each part of the body is capable of dividing into an adult, and this process is known as regeneration. Okay, uh, here I have given couple of examples. Like first one is of a flatworm planaria. If accidentally the body of the planaria divides into two, each part can is capable of developing into an adult. Similar thing can happen with the body of hydra also, and a starfish, but only the arm of the starfish is capable of regenerating, not any other part of the body. Okay, so suppose the central disc of the starfish is removed, and then this arm will be capable of developing into an adult. this will be a difficult process. Okay, so starfish also, they are capable of regenerating. There are some starfishes that can regenerate also. Their arms also can regenerate, which specifically is shown in this diagram. Okay, so regeneration is more like, you can say, it is a process of survival, right? This is how the animals, they can survive if there is an attack by a predator, or the conditions are harsh, high tide, or storms, or anything like that. Okay, so regeneration is also common in lizards. Here they can basically regenerate and regrow their tail. So they have certain cells inside their tail which can regrow and regenerate. Okay, in human beings also, there is an organ which is capable of regeneration, and that organ is liver. So this is a new information, I think, for you guys. Livers, that means they are the easiest organ that can be found if organ donation is required. Okay, so regeneration also is done. With this, we are going to start with the questions. Okay, in the first question, you have to identify the processes given in the following figures. Okay, so the first figure is of multiple fission, right? If you're probably thinking multiple fission, then you're correct. I'm writing the examples also in the bracket. This occurs in Plasmodium, and this is regeneration, and regeneration occurs in starfish. Okay, let's move on. The eggs of frogs do not have shell for protection, yet they are safe in water. How? Yes, that is true. Eggs of frogs do not have shells. Okay, the eggs have jelly-like substance covering them. Okay, and this jelly-like substance, it keeps them moist and prevents them from desiccation, prevents them from drying up. If the frogs lays eggs on land, they will get dried up and die. Okay, this is how they are safe in water. Okay, the egg nucleus is not exposed to the water just like that. It still has a covering which is jelly like. Okay, which protects them. The next question is about twinning, how twinning occurs during sexual reproduction. Okay, twinning is the formation of twins. Okay, so there are two types of twins. There are identical twins. Okay, and there are also non identical twins. Okay, so let's look at the picture to understand the identical and the non-identical twins. In the identical twins, the fertilization occurs and the zygote develops. And this zygote, remember I told you that zygote continuously undergoes division, it divides and redivides. So while one cell is dividing into two cells, these two cells accidentally separate and they are implanted separately. Okay, so when the two embryos, two zygotes are coming from the same cell, it is known as identical. It results in the formation of identical twins, and they are known as monozygotic twins. Okay, but sometimes what happens is uh, eggs that are coming out from both the ovaries, usually every month only one ovary produces eggs, but sometimes accidentally both the ovaries, they produce eggs in the same month, and fertilization takes place. Both the eggs get fertilized and get implanted in the uterus. Okay, simultaneously. This is how the dizygotic twins are produced, that are non-identical twins. Okay, the sex of the zygotes can be same or it can be different. Okay, so the monozygotic twins will share the same placenta, but the dizygotic twins, they will have two placentas, like there will be two umbilical cords in case of dizygotic twins, and there will be only single umbilical cord in case of a monozygotic twins. If you are confused about umbilical cord, it comes out from the placenta of the mother, and then it connects to the womb of the baby. Okay, so this is how the twins are produced. Let's move on to the next question. So visit a poultry farm, talk to the manager of the farm, and try to find out the answers to the following. What are layers and broilers in a poultry farm? Layers are the birds which are grown for especially only for eggs. Okay, and broilers, these are the eggs which are grown only for their meat. Okay, so layers can lay good quality of eggs. Okay, and broilers, they can give good quality of meat. So this is egg laying birds. Okay, do hens lay unfertilized eggs? Yes. How can you obtain fertilized and unfertilized eggs? Fertilized eggs can be produced by natural process. Okay, naturally this can happen, but unfertilized production of unfertilized egg is induced by hormones. Okay, hormones are injected into them, and then the eggs are produced. Are the eggs that we get in the stores fertilized or unfertilized? So the answer for this is both. Both type of eggs are found in the stores. Can you consume fertilized egg? Yes. Is there any difference in the nutritional value of fertilized and unfertilized egg? Yes, there is. Fertilized eggs are basically more nutritious, okay, than the fertilized, sorry, than the unfertilized eggs. Unfertilized eggs are also nutritious, okay, but if you compare the nutrition value, fertilized eggs will be more nutritious. Let's move on to the next question. Internal fertilization takes place inside the male body, inside female body, outside female body, or outside male body. The correct answer is inside the female body. This we have already discussed. It is the fertilization that takes place inside the body of the mother. Okay. Next question is, sets of reproductive terms are given below. Choose the set that has an incorrect combination. Okay. So first is sperm, testes, sperm duct, penis. B, menstruation, egg, oviduct, uterus. Okay. C, sperm, oviduct, egg, and uterus. Then D, ovulation, egg, oviduct, uterus. The correct answer is C, where sperm, it reaches the oviduct where it meets the egg, fertilization takes place, and finally the fertilized egg is implanted into the uterus. Okay. Out of all the options, only C, option C makes sense. Okay. Then, which is not a viviparous animal? The viviparous animal are those which give birth to young ones. Human beings do, cows do, dogs do, but butterfly does not. So the correct answer is butterfly. Okay. So, guys, we have finished the topic with this question, and we have finished the video also. I hope you liked it, and more than that, I hope you understood it. I would love to answer your questions. If you have any, you can post them on the forum, and stay tuned for my next video on next topic. Thank you.